Second part for that chapter 26, we're going to look at here the structure and the function of nucleic acids. Well, looking at here the sequence in which the nucleotides are connected is very important. And if you look at this, well, the sequence of nucleotides in the nucleic acid chain is read by starting at the 5' end and identifying the bases in the order they are appearing to. So look at here on the right, 5' position, you have thymine, you have some phosphate diester links, then you get adenine, and then you get guanine. Well, what is very interesting is that studies from Chargaffs, who was a very important scientist, look at if any given species, the amount of adenine and thymine was always equal, and separately the amount of cytosine and guanine was also equal. So that is what we call Chargaff's rule. So it tells you that the amount of adenine and thymine were equal and amount of cytosine and guanine are equal. So A equal T and G equal C. So that will really help to understand the structure of that DNA molecule, and then that suggested that the bases occur in discrete pairs, and that's here where the proportion of each A to T and G to C vary from one species to another. Very important, 1953, Watson and Crick, who get a Nobel Prize, propose a structure of the DNA, and that will be accounting for that pairing of the bases and also the storage and transfer of the genetic information. That's why we call the double helix. That's the Watson and Crick model. Double helix, you have two polynucleotide strands that are coiled around each other in a helix, so helical screw-like fashion. And then the backbone is made of sugar phosphate, that's on the outside part, and the right inside is actually taking inside those bases, nitrogenous bases. So looking at this here, you look at the base pairing, the Watson-Crick model. So the right side, this is the double helix. Now in the middle, it shows you that one strand runs from 5' on the top, left, to 3' on the bottom. The other one is opposite. We say they run in anti-parallel directions. And then if you look at the left side of this figure, you see that T has a double bond with A. Well, actually, those are hydrogen bonds. So there are 2 links for T and A, and there are 3 links of G and C. Very important to remind them. And remember also that T always pairs up with A and G always pairs up with C. That's what we have right there, 2 hydrogen bonds for A and T and 3 hydrogen bonds between cytosine and guanine. So pairing of those bases is said to be complementary. Every time you have a thymine on one strand, the other one will have adenine. Every time you have cytosine on one strand, the guanine is opposite on the other strand. Now A and T and C and G. Now remember, when we look at RNA, there is no thymine that is replaced by uracil. Very important, this is why we have DNA. That's your heredity as long as, actually as soon as you have a fertilized egg. And that's from that egg that you're going to grow. So you get, we said, 23 pairs of chromosomes, so 46 chromosomes total for humans. You inherit 23 chromosomes from your dad and 23 chromosomes from your mom. Other thing important, well, we're going to look at here heredity and cell division. This is an ongoing process. No single cell has a lifespan equal to that of the organism. So every time a cell divides, the DNA must be copied. We call that replication. And looking at the double helix of DNA and the complementary base pairing make that duplication possible. Every single strand of the double helix is what we call the blueprint of the other strand. And we're going to look at here, well, genetic information is conveyed not just in the numbers and kind of base in DNA, but also the sequence that exists along the bases and the DNA strands. Sometime we'll see later that sometimes mistakes do take place, and that's going to actually have what we call mutation. Genetic information encoded in the DNA directs to synthesis of protein, which is what we call the expression of gene, and that's the second part of that lecture.